Hello, BIS 210 students. Today I am going to be going through DNA replication, followed by gene expression, transcription, translation, and then I'll talk a little bit about operons. So first things first is we're going to discuss DNA replication. DNA replication. Now, sorry, I have horrible handwriting, just the way it is. Now, in DNA replication, keep in mind, bacterial cells only have one chromosome. It's circular. And there are two replication forks. So if we were to draw it out, you would have circular chromosome, and you'd have one replication fork going this way, one going this way, and they would meet right here along the middle as they go. And once they reach this spot where the green line is, that means they've copied the entire chromosome, so what they end up with is a second chromosome. So here's number 1, and here's the identical number 2 chromosome. So this is essentially what binary fission is, but we have to copy the DNA first, right? This process here, replication, has to happen before binary fission. Because the point of binary fission is to create two new daughter cells that are genetically identical. Well, in order to have genetically identical cells, you've got to copy the DNA exactly. And that's what DNA replication is. Now, what we're going to do is we're going to focus in on one of these forks, whether it's this one here or this one there, doesn't really matter. We'll focus on one of them. But just know, even though if we focus on one, it's happening in the opposite direction as well. So we'll look at one of these, and then you just need to know that it's happening in both directions. So first and foremost, we're going to draw out our DNA. Now, if you recall, DNA has directionality. There's a 5' end, and then it has a 3' end. And then the opposite, right? So 3', 5'. So the top strand is 5' on the left, 3' on the right. The bottom strand is 3' on the left, 5' on the right. If you recall, there's a very specific reason why this has to happen, why the strands have to be opposite of one another. And hopefully you remember they're anti-parallel because of the nucleotide structure. So before we even start replication, let me go in and show you, or remind you, what the structure of a nucleotide looks like. Okay, you have a phosphate backbone, you have a sugar, and then a nucleotide or a nitrogenous base, like adenine, guanine, cytosine, thymine. Right? So if we're looking at one strand and they all line up like this, that's perfectly fine. Right? We want them to be in a line. But what happens if the opposite strand essentially is in the same direction? So let's say this is 5, this is 3, and you have your phosphate, your sugar, and your nitrogenous base, or nucleotide. Phosphate, sugar, nucleotide. If this other strand is in the same direction, there is a problem. It would be phosphate, sugar, nucleotide. Phosphate, sugar. So they would be, instead of the nucleotides facing each other, this nucleotide on the left strand would be facing the backbone or the phosphate of the other strand. So there's a really easy way to fix this. Just flip one of the strands. So if we flip one of the strands and if we flip one of the strands and make it 3' to 5', the opposite direction, phosphate, sugar, nucleotide. Phosphate, sugar, nucleotide. And then these will interact with each other. Here's your other strand. So you can see by flipping the directionality of one of these strands, the nucleotides will face each other. And now DNA is happy because the nucleotides are interacting. Okay. This is referred to as anti-parallel. Strands are in opposite directions of one another, which is very important. And it's going to be important when we talk about replication as well as transcription and translation as well, because directionality is important to certain enzymes. All right, so let's get back to this. This is our bacterial chromosome, but notice what has happened. Notice what's happened. There has been an enzyme that has unwound the DNA. DNA is in a supercoil, right? A winding staircase. So there has to be an enzyme that unwinds it. Gyrase. Followed closely behind by the enzyme that unzips the DNA, helicase. So gyrase unwinds, helicase unzips. Now what do I mean by unzips the DNA? Well, what I mean is what holds the strands together. So you have these nucleotides in this region right here. What's holding these nucleotides together? What's holding the nucleotides together? We talked about this when we talked about chemistry, and we also talked about this, I mentioned it in the lecture video. So DNA strands are held together via hydrogen bonds. So the hydrogen bonds between these nucleotides, they're still intact, but the ones over here to the right, where the replication fork is, they're separated because helicase has broken the hydrogen bonds, separating them from each other. Now, a problem here. DNA dislikes very much being separated. It does not like being open like this. So there are these proteins that will attach to the outside of the strands called SSBPs. And it's these proteins, same thing with the ones on the bottom strand. SSBP stands for single-stranded binding protein. Single-stranded binding protein. And their job, essentially, is just to hold this open. Because if they're not there, DNA closes. And if DNA closes, then it can't be copied. So, single-stranded binding proteins hold the strands open that allow the enzymes to get to each strand and build a complementary or matching strand. So, how are we going to build the new strands, right? That's our next step and our next goal. Well, we're going to use both the top and the bottom strand. We're going to use both because remember, we're making two copies. So we need one full copy to go to one cell and another full copy to go to the other. So we have to use both strands to make new, we have to use both original strands to make two new strands. I'll show you how that works. So we're going to look at the top strand first because this is what we call the leading strand. The leading strand. Because it's really easy to for the enzymes to do their job because there's nothing in the way. So what happens is there's an enzyme called primase that puts down a little section called a primer. Okay? The purple thing is a primer. Okay, it's called a primer. Now, the primer's function is to tell the DNA polymerase where to bind. Now, polymerases, there are multiple types in your book. It'll tell you there's like 3 or 4 different types. I don't need you to know the difference between them. I just want you to know what polymerase does. Polymerase is what builds the new strands. It is the enzyme that builds the new strands. Now, we also have RNA polymerase, or pseudobacterial cells, but we're using DNA polymerase in this case. And the answer to the question as to why that is because what is our product? Are we making DNA or are we making RNA? Well, we're making a DNA copy of these strands. So since our product is DNA, we're going to use DNA polymerase. And here is DNA polymerase. DNA polymerase always reads in the 3' to 5' direction, and it synthesizes in the 5' to 3' direction. So this thing is going to read this way, because it's going to read 3' to 5', and it's going to read that top strand, and as it reads the top strand, it is going to build a new strand to go with it. It's going to build an identical copy. What we call a complementary strand. Okay, so it's going to build a complementary strand. And since the top strand is 3' to 5', the strand it's making has to be 5', and this is the 3' end. The 3' end. So hopefully this makes sense so far, that we have used the top strand to use one of the original strands, the top one, to build a new daughter strand to go with it. But that's not enough, right? That's only one copy. We need a second copy. So we will use the bottom strand as well to make another complete copy of the DNA. Now here's the problem. Look at the directionality of this bottom strand. You have 3' here, 5' there. We know polymerase reads in the 3 to 5' direction. With this section here closed, no enzyme, meaning polymerase, can get access to this. it can access this section, but it can't access this one. So the bottom strand, we have to build it in pieces. We have to build it in pieces or chunks. So what we do is very similar to the top strand, is we put down a primer, and it's actually what we call an RNA primer. Put down a primer, DNA polymerase binds, and it builds a section. It builds one section. Because remember, this has to go it has to read the bottom strand 3 to 5. So it's going from left to right, where the top one is going right to left. So it builds this little section, or what we call a fragment. This fragment right here has a special name. It is called an Okazaki fragment. Okazaki fragment. Basically, the bottom strand is made up of a bunch of Okazaki fragments. It's being built in pieces, and then it's being tied together to make one continuous strand. So, let's get rid of the little box, and we're going to say polymerase is gone as well. Polymerase is gone as well. We're left with our primer and then the section that it built. The problem is that primer needs to leave. That primer needs to go away. So the next fragment that's being built, the next Okazaki fragment, that polymerase will pop, it'll get to the primer, and it will pop the primer off, it'll remove it, and it will fill in the gap where the primer was. However, there is a catch. The polymerase can put the nucleotides, whatever nucleotides they are, adenine, cytosine, guanine, thymine, whichever, they can put the nucleotides in place, but they don't have the ability to tie them together covalently. So there's another enzyme that comes in called ligase. Ligase. When you ligate something, it means to tie. So what it does is, we filled in the gap, right? What it does is it ties the pieces together. So now you get one continuous strand. And then this will happen all over again. Primer gets put down. Polymerase builds this next section. Gets to the old primer, removes it. Removes it, fills in the gap, and ligase comes along, ties it together, so it's now one continuous strand. So you can see how the bottom strand is built in sections. We call that the lagging strand. And the top one is called the leading strand. So you have the leading and the lagging strand. Now I know I'm throwing a lot of enzyme names and functions at you, but make sure you listen to this video a couple times and kind of go through each piece one by one. One way I suggest you practice this is to draw it on your own. take the template that I use, the black lines, right, the strands, open it up at a replication fork, and draw in what you expect to be there and why it's there. Okay, so we're still not done. This is just one of the replication forks. The exact same thing is happening on the other side where the other replication fork is. And then they eventually meet in the middle. And then they have two copies of the new DNA. So what you have, my question to you actually, is what do you have as your final products? Do you have this? Do you have this? Or do you have, oops, or this? So think for a second. After you've gone through DNA replication, you have two circular chromosomes. What would they look like? What would they look like? Hopefully, you selected C. Because remember, we're using one original strand. If we go back, we're using one original strand right there, and we are building a new strand to go with it. Same thing here. Original strand, new strand. So what it actually looks like is this. You'll have to excuse my terrible artistic abilities. You have one copy. Here's your first copy. Here's your second copy. They have one original strand and one new one. One new strand, one original strand. This is referred to as semi-conservative replication. This is called semi-conservative DNA replication. Now, what does that actually mean? Well, semi-conservative is referring to the fact that you use an original strand, but you also pair it with a new strand. If something was completely conservative, you would only have the original parent strands. If something was not conservative at all, you'd only have new strands. But if you are semi-conservative, you have a mixture. You have one original strand matched with one new strand. And that's what you have here. That's what you have in this. So there's your products of DNA replication. These are your products. And this one, copy 1 will go into cell 1. Copy 2 will go into a different cell. And then at the end of binary fission, they split, and you have two new daughter cells. And they're genetically identical. There could be possibly spontaneous mutations or random mutations, but they're pretty rare. They're pretty rare. So I wouldn't bet on it, but genetically they should be identical, unless some mutation happened somehow, some random mutation. So this is DNA replication. Okay, this process is completely separate from what we're going to talk about next. This process, DNA replication, its goal is to copy the genome exactly all the genetic material, copy it all, and have one copy go to one new cell and another copy go to the other new cell, which are created through binary fission. So all this has to happen first, and then binary fission occurs. So they copy the DNA, make 2 copies, and then you have binary fission happen. You then have binary fission happen. And each daughter cell gets an identical copy of the genome. Alright, so this is DNA replication. If you have questions, make sure you ask me during office hours or during SI, ask your SI leader. This is an important concept. And we'll talk about gene expression next, which is just as important, if not more. Okay, so next we're going to talk about gene expression. Gene expression. So we know that the central dogma is DNA, RNA, specifically mRNA, and then protein. This is how the genetic flow of information occurs. So to express a given gene, so let's think of DNA as a cookbook, right? I kind of gave this earlier in the semester, right? DNA is our cookbook. We want to, a gene is a single recipe. We want to make that recipe, which eventually is, i.e., a protein. But there are 2 steps that have to happen in order to make this a reality. You have to have transcription, between DNA and RNA. So you transcribe DNA into an mRNA copy, and then you have mRNA, and it undergoes translation to make up a functional protein. So this is to express genes. Now it could be any gene. It could be a gene that causes pathogenicity. It could be a gene that's associated with the membrane. It could be a gene that makes arginine, which we'll talk about arginine later. It could be anything, really. But it's still going to follow this process. DNA is going to be transcribed to mRNA. mRNA will be translated to a protein. So let's talk about transcription. So we have our DNA, and I'm going to draw it out. Here's our 5' end. There's the 3' end. Here's the 3', 5'. Okay. So what we do, or what bacterial cells do, is they open up these little bubbles in order to transcribe the genes. Because remember, the gene, one single gene does not take up the whole chromosome. It's just somewhere along the chromosome, right? So let's say, I will promise I'll go back to that slide in a second. Let's say, here's the bacterial chromosome, and right here is your gene of interest. That's the one you want to transcribe. So we would open this region up. And that's exactly what's happening here, is we're opening this region up. Now we're going to see a lot of the same enzymes that we saw before with replication. Up here you have gyrase, unwinding. You have helicase, unzipping. You don't use SSBPs here, no single-stranded binding proteins, but we've used gyrase and helicase. Now, when it comes to the transcription process, we use another polymerase. And again, there are multiple polymerases. However, I don't expect you to know them all. We're just talking about them in general. What did we say about which polymerase we use in which process? So in DNA replication, we used DNA polymerase, but why? Why did we use DNA polymerase in DNA replication? Because our product was DNA. In this case, for transcription, our goal is to make an mRNA copy of the gene. So our goal is to make RNA. So we would use RNA polymerase. Now, the polymerase doesn't know exactly where to bind without looking for a specific region. There's a specific region called the promoter region. The promoter region. This tells the polymerase where to bind. And we are using RNA polymerase. We know polymerase reads 3 to 5, so we're looking at the bottom strand. And it's going to go from left to right. Now the same thing is happening on the top strand, but we're only looking at one. It's important to know, though, whatever we're doing on this bottom strand, this whole process that I'm about to draw out, is happening on the top strand as well at the same time. Because if they have to make a ton of, let's say, arginine, right, it makes sense that they transcribe both the top and the bottom strand because, well, they're complementary, they're matching. So you would still get arginine, right? So it's kind of important to understand that bacterial cells and human cells, for that matter, have multiple replication sites, or transcription sites, excuse me. So here, we're going to focus on the bottom strand, but the top strand is also doing the same thing, just in the opposite direction. So polymerase is reading, RNA polymerase is reading left to right. So what it does is, as it gets over here towards the bubble, it starts putting down matching nucleotides. And it builds the polypeptide. So let's say it's over here. Still going that way. So it's building the mRNA strand. So it's building the mRNA strand. And it's going to keep going and building a complementary or matching mRNA strand for as long as the gene of interest is. So if it's 500 nucleotides long, it'll transcribe 500 of them. If it's 5,000, it'll transcribe 5,000. Okay? It will transcribe the whole thing. So, now, once we have this mRNA strand being built, at some point, the polymerase is going to hit what we call the terminator region, which tells it where the end of transcription should occur. So let's say it's all the way over here, this brown region right here is the terminator. right there. So it will transcribe everything up to that point. So everything between the promoter and the terminator, almost everything, will get transcribed. And what you end up with is an mRNA strand that looks like this. And then you can just now the way I'm drawing this is a little different than what it looks like in real life. Okay, there are no gaps. So notice how I drew them in triplets. There's a very specific reason for that, which we'll talk about in a second. But normally, there would be no gaps. There would be nucleotides jam-packed in this whole thing. So, for instance, like this. The whole thing would have nucleotides. Okay? But, for our purposes, I separated them out for a reason. Now, we have our mRNA copy of the gene. We need to know what to do with it. All right, we need to know what to do with it. Well, the next step is translating it into a functional protein. So how do we do that? Well, instead of using a polymerase, we actually use a ribosome. And bacterial cells use a ribosome, so do humans. Use a ribosome to translate. So the reason I drew them out in triplets is because that's how a ribosome reads the mRNA sequence. It reads it as 3 nucleotides equals 1 codon, and 1 codon codes for 1 amino acid. So 3 nucleotides equals 1 codon. 1 codon codes for 1 amino acid. So each triplet codes for 1 specific amino acid. And I talked about triplets a little bit when in the video lecture when I talked about that chart with the triplet sequences and how you could have serine, the amino acid serine, coded for 6 different ways, and leucine coded for like 6 different ways, and all these others that code for multiple are coded for multiple ways. So it's important to remember that 3 nucleotides equals 1 codon. 1 codon codes for 1 amino acid. But let's say you have CCC and then CCA as the first 2 codons. They could code for the same amino acid, right? Leucine's coded for 6 different ways. So technically, they could be coded for the same way, even though the codon, or the actual sequence, is not the same. One of them is CCC, the other is CCA, right? They're different, but they still code for the same amino acid. There's a lot of redundancy when it comes to this. Okay, lots of ways to code for the same amino acids. Okay, so the ribosome is going to attach to the mRNA strand in 2 pieces. It has the top portion, the small subunit, and then the bottom portion, which is the large subunit. Essentially, it clamps down. Imagine, a rope, and all of a sudden you just grab it with your hand and wrap your hand around it. Okay, You've literally clenched that rope in your hand. That's what's happening with the ribosome. It is joining together, clamping down on the mRNA sequence. Now, from the video lecture, I told you it's looking for a very specific sequence to start. So when I showed you that table with all the different codons, I told you only needed to know 4 of them, right? Only 4. The start codon, which is AUG, and it codes for methionine, and then the 3 stop codons, UAA, UAG, and UGA. So we have AUG right there. There's our first one. Now, it doesn't matter what the next ones are, so I'm going to write CCC, ACC. Okay, we're going to do that. Now, we need to build the polypeptide, but the ribosome can't be attached to the mRNA and still go and grab the correct amino acid and bring it back, right? It's kind of silly to think that, okay, well, it latches on and it's reading the sequences, and then it lets go to go find the amino acids and bring them back and latch on again. That makes no sense. It's wasted energy. So instead, it uses another type of RNA molecule to help it build the polypeptide. Now, I promise I'll come back to this slide in a second. The molecule that it uses is tRNA, or transfer RNA. So this is tRNA, transfer RNA. Transfer RNA is really interesting because it has an anticodon on the top. It has a really weird structure, and then on the bottom it has the correct amino acid, or so let's say it's just an amino acid. It has the correct amino acid. So it's got an anticodon on one end, and then the correct amino acid that matches it on the other. So if we're to go back to that table with all the codons, if this anticodon was CCC, it would code for a very specific amino acid. So the anticodon codes, it codes for the correct amino acid based on that chart. Now that chart was an abbreviated version. There is actually a larger one, but it gives you the general idea. So the tRNA is what's going to bring the correct amino acid to the ribosome so it can build the polypeptide. So if we go back here and we say, all right, this is green. Let me change the color of the ribosome. Okay, let me just change the color of the ribosome so I can use green. Okay. So the ribosome's attached. It's looking at the first codon, and it's AUG, the start codon. What is the anticodon to this? What would be the matching codon that would pair with this? Because it's going to call the tRNA over, and it says, hey, the tRNA that has this anticodon needs to come over here. What would be the matching sequence with AUG? The matching anticodon. I'm going to give you a second to think about it. All right. Well, we just think about what pairs with what. Adenine pairs with what? Now be careful. Got to remember what we're looking at. We're looking at an mRNA sequence. So if we're an RNA, it's not thymine, it's uracil, and uracil and adenine pair, and guanine and cytosine pair. Oops. That should be cytosine, I apologize. Cytosine. There. And there's your anticodon, the weird structure of the tRNA, and it brings the first amino acid, which is methionine. Methionine is the start codon. That is the first one you will see. Okay, the anticodon is UAC, and it codes for methionine. Now, it still triggers more tRNA to show up. All right, the next one is CCC. Well, the anticodon is GGG, right? tRNA has a weird structure, brings the next correct amino acid, and then they are covalently bound together. So a covalent bond forms between them. This third one, it's ACC, so it's UGG, has its weird structure, and the next amino acid. And then it's covalently linked. And the ribosome will read this way, from 5 to 3. So the next thing it does is it just scoots over a couple codons, or one or two codons, and everything shifts. So when the ribosome moves over one, this first tRNA leaves, and it goes and picks up another methionine. So let's draw that out. So we have our 5 prime, 3 prime. We have 1, 2, 3, 1, 2, 3. Okay. So the ribosome shifts over one spot. Here's where AUG was. There's AUG. The ribosome shifts over a spot, and all of the tRNAs will shift as well. So we had what? CCC, ACC. So we had CCC, ACC, and then let's say AAU. And we had our tRNAs in here. We had our methionine. We had the next amino acid right here, covalently bound. Here's the UGG. The next one, the correct amino acid, covalently bound. And then another tRNA shows up, UUA, weird structure, and the next amino acid. So you can see how as the ribosome shifts over, it boots out. So if we say this is position 1, 1, 2, 3. And they actually do have names. They have the what we call the EPA sites, but we're just going to call them 1, 2, and 3. Positions 1, 2, and 3. As this moves over, the tRNA in position 1 will exit the ribosome, leaving the amino acid, and it will float away and pick up pick up another amino acid. Pick up another amino acid. And it can be reused. So tRNA is not just used once and thrown away. It's reused. So as this thing shifts, this first one gets booted. The second one moves into position 1. The third one moves into position 2. And then a new one will move into position 3. So you can see how everything moves over one spot every time the ribosome reads the next codon. Okay, until eventually it hits a stop codon, which hopefully you remember. UAA, UAG, UGA. These are the stop codons. They do not code for a specific amino acid. All they code for is the ribosome to stop translating. So eventually it'll hit one of these, it'll hit one of them, and that tells the ribosome, all right, you're done. You've translated the entire protein. Now this could be 50 amino acids, could be 500, could be 5,000. It really depends upon how long the gene is. So what you end up with, your product is methionine, and then a bunch of amino acids put together in a chain and covalently bound. Covalent bonds between them. This is your product. And this is the primary protein structure, where it's just a chain of amino acids. This is the primary protein structure. Now, hopefully, you remember from earlier chapters that proteins are not functional when they're in their primary stage. I know I didn't go into it a lot, but I told you that proteins have to do something very specific. They have to fold. So, yes, this is the primary structure, but the final structure of the protein will have a distinct shape. So it could just be, it looks like that. It folded into, this is the correct shape, and then it works. It works. But if it doesn't fold properly, notice it's missing this group right here and this group right here. So notice this is not correct. And does not work. Okay? The protein will fold to a very specific shape. When it folds to that shape, it becomes functional. If it does not fold to the correct shape, it does not work. If it folds and it's very, 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 very close to the correct shape, the protein might function ever so slightly, but in most cases, if it does not fold to the exact correct shape, it will not function properly. It will not function properly. So that is gene expression, creating a protein and going through transcription and translation. Okay. If you have questions, please make sure that you ask myself or your TA, or I'm sorry, not your TA, your SI leader. Don't ask your TA, ask your SI leader. Or come to my office hours. Now, there is one more thing I want to cover. Okay, so there's one more thing I want to cover, and then I will answer any questions you may have. So it kind of ties in together, gene expression. Do we want genes to be expressed at all times? The answer is no, right? Sometimes you have enough of the product where you don't need to have genes being expressed any more than they already are, or they need to be turned down. So remember, we have a bacterial circular chromosome, and they have genes grouped together in operons because they get transcribed and translated together because they're similar in function. They are similar in function. So maybe these ones right here are membrane proteins. These ones are lac operon, breaking down lactose. These ones are pathogenicity genes, genes that make the cell be able to cause disease. So they group them together based on function and the fact that they get transcribed and translated together. But don't always want to you don't always want to have these genes being expressed at all times. Okay, you need to be able to regulate gene expression. So in the chapter, I talked about the arginine operon. I mentioned the arginine and how it's repressible, meaning it's always on. I want to show you another example, the lac operon. The lac operon is pretty simple compared to some of the other ones, but let's talk about the lac operon, what we call the lactose operon. So we call it the lac operon. So here is your DNA. Here's your 5' end, here's your 3' end. Now you have some distinct sections in here. You have gene 1, gene 2, gene 3, and then in front of that you have the promoter and the operator. The promoter and the operator. And then this is just more DNA down here. So, the promoter and the operator, if you recall back to what we just covered, promoter tells the polymerase where to bind, right? So if we're going to transcribe these genes, right, we're going to make mRNA, therefore we're using RNA polymerase. So the promoter tells RNA polymerase where to bind. And if there is a lot of lactose in the cell, this will transcribe those genes, because these genes, all three of them, they're enzymes that break down lactose. They're enzymes that break down lactose. So if lactose concentration is high, you will get transcription and translation of these three genes. So it can break down the lactose in the cell. If lactose concentration is low, is there a point to making these 3 genes? Is it necessary to make genes, 3 of them, to transcribe and translate them when they don't have a ton of lactose in the cell? The answer is no. It's not necessary. So to save energy, the cell can actually block transcription. And it does so using what we call a repressor. Repressor protein. The repressor protein attaches to the operator region, and it literally blocks the polymerase from going any further. It can't get past the repressor. So what's happening then is these 3 genes, this one, and this one, are not getting transcribed and translated because there's not enough lactose. So we don't need them, right? Bacterial cells don't need them to be transcribed and translated. So the repressor binds to the operator. And then lactose concentration will increase, and once it gets high enough, the repressor will let go. Lactose will actually bind. So let's say this is lactose. These are lactose. And the concentration now is really high. What'll happen is one of them will bind to the repressor, and it will let go of the operator. It'll let go of the operator. And polymerase now has the ability to transcribe and then eventually translate those 3 genes. So it's not a permanent turn on or off. It can be turned on and turned off and turned on and turned off over and over and over again. So what determines whether it's turned on or off? Well, lactose concentration. If the lactose is high, then you get transcription. If lactose is low, you do not get transcription. Okay, depending upon the lactose concentration, will determine whether or not the repressor binds to the operator and whether or not transcription and then translation will occur. So it's all about lactose concentration. Please, if you have questions about this, let me know. So I was asked by several students to cover one more topic in a little more detail. I had intended to stop at the operons. However, I was asked by several students to kind of talk about mutations a little bit. So let's talk about mutations for a second. So we have our sequence. Here's our sequence. Now let's break it up into codons. Dog ate bat and hat. Okay, obviously these are not real codons, but we're going to use real words because it's actually a little bit easier to to see what happens. So, all of these code for a very specific amino acid, but let's say a substitution mutation happens, and you end up with dog ate cat and hat. So there was a substitution at this specific location. So the amino acids here, it's the same as the one here. This one's the same. This one is the same. This one is the same. None of those were affected. This one, the one that has the change, there's three possible outcomes. There are three possible outcomes. First, a silent mutation where the amino acid does not change. So let's say bat codes for serine. There was a substitution and now it says cat, and it still codes for serine. We call that a silent mutation because who cares, right? It still codes for the same amino acid. That's all that matters. Or you can have a missense, which is the amino acid changes. The amino acid changes. So if that right here codes for serine, and then you get this specific mutation, now it codes for proline, the amino acid has changed, therefore it is called a missense mutation. And then the third thing that can happen is a nonsense mutation. And this is where you actually code for a stop codon. So if this change led to a premature stop codon, the whole protein wouldn't get made. You'd only make half of it, the first half. You'd make this one and this one, but if this change triggered a stop codon, this wouldn't get made and that would not get made either, and anything else down the line. So a premature stop codon is called a nonsense mutation. These are all due to a substitution mutation. One nucleotide is substituted for another. So these are the three outcomes that can happen. Now, one more thing is if we have dog ate bat and hat. Dog ate bat and hat. If we were to have a insertion or deletion of a nucleotide, it changes the way we read the sequence. So let's change that, right? So let's say this B gets eliminated. Now everything on the right shifts to the left one spot. So now the way we read the sequence is dog ate ATANDHAT. This one and this one remain unchanged, but everything else is now different. It changes the way we read the sequence. So in a deletion, it shifts over to the left, however many are deleted. So since we only deleted one, everything on the right side shifts to the left one spot. And it changes how we read the sequence. If you get an insertion, so Doug 8, and then we'll add, instead of deleting, we'll actually add another letter, or another nucleotide. So we'll add an A. But now everything has to shift over to the right. So it will be A, B, A, T, A, N, D, H, A, T. This one and this one, they're fine. They're fine. But everything else after it shifted to the right because we inserted an extra nucleotide. Where in this one, we deleted one. And everything shifts, in this case, that way. This is called a frame shift mutation. A frame shift mutation. And that is because it changes in which, the changes lead to how you read the sequence. So what we call our reading frame shifts. Our reading frame shifts, and the way we read the sequence becomes very different. And frame shifts, most of the time, are going to make no sense. So when everything shifts, essentially that protein is useless. Because now the way you read it is very different, and a lot of the amino acids that are coded for are going to change. So frame shifts are pretty drastic and usually lead to non-functional proteins. Okay, so I was asked to go over those types of mutations. That's all I have to cover for this chapter. I know this is a little bit longer chapter, which is why I allot 3 class periods for it, but I still went a little bit above that. However, like I said earlier in the semester, there are going to be times where I'm under and a few times where I'm over. So it all evens out to the correct number of, in terms of time, classroom time, so to speak. This one's a little bit over. But it's still very, very important that you understand what's going on. So please, if you have questions, talk to your SI leader. Go to SI. Come to my office hours on Wednesday, Thursday from 12 to 2. I'm happy to answer any and all questions. All right. I hope everyone has a nice day, and I will talk to you all soon. Stay safe.